Hey class, let's look at, so remember last time we talked about the peptide bond. So it's simply the peptide bond is this little dash here. That's a C double bond ONH bond, which is a covalent bond, but it's not a simple line just like between H and Cl, between carbon and carbon, right? But the other thing also that this look at this one, lies ala, his, Clay, and so on. This sequence, the order is actually, we read it from N terminal, if you remember N terminus, and then that is the amine group sticking to the left side of the screen, and then the right side of the screen, which is the carboxy end, right? So that's important to understand that when we read the peptide sequence, the primary structure, we read it N terminal to 5 terminal, sorry, N terminal to carboxy terminal, right? And so it's similar to when we read the genetic coding, we read from 5 prime to 3 prime, right? The coding strand. So same style, right? Okay, so from there, I want to look at that bond that I was talking about. This is the peptide bond. So the peptide bond here is actually, you have a couple of things. See, here is the C alpha of the amino acid 1. And here's the C alpha of the amino acid 2. So now the amino acid 2 and the amino acid 1 come and combine, right? When they combine, that makes the peptide bond. So now there's another thing here. So yeah, looks like my drawing is gone. So let me see whether I can put it quickly here, right? Yes, so I do have, this is the C alpha of the amino acid 1, and here is the C alpha of the amino acid 2. And then what happens here is actually there is a lone pair. If you are aware of the lone pairs and number of electrons, it's easier. But otherwise, I would just memorize what the picture is, right? So this goes here and this goes there. So therefore, I get an extra minus, so that means it's actually something like that. And this is a positive on the nitrogen, negative there, and this is a double bond, right? So now what actually happens, this is between the two resonance structures, if you remember. And these two resonance structures are in between, right? So they are not really either left or the right, it's in between. The real thing is in between. So these two drawings are only on paper, right? It's theoretical. So in reality, what we have is in between about 40% of the double bond character. And so that gives us another thing. So this whole thing, the all 6 atoms are in one plane. So I would simply say it is, the angles are 120 degrees, right? 120 degrees for each and every bond in this case. And it's planar. So it's planar and has a 40% of double bond character, and it's planar, right? So that kind of we want to understand, because that's very special shape it gives you, right? The result will give you. Okay, now this is another way. It's a little complicated, so I wouldn't worry too much about the 116, 119, all the details, but I would simply say this is sp2 hybridization and partial double bond here, and 120 degrees roughly for each of them, and then the C alpha here for the second amino acid, C alpha here for the first amino acid. So all these 6 atoms are in one plane, so amide plane, right? So that we need to know. Now I have a bunch of those planes, like my playing cards from my kid, remember? So those ones are actually stick together with a little tape that I had. And so that's just like a really good way to think about the sequence. And then look at this one. We also call, let me go back a bit. So this one is called the trans peptide bond. Because trans is like, see, if it is a double bond, we know it's partially. And then see, this R group is sticking down and this R group is sticking up, right? So therefore, it's a trans. So it's a good way to explain. Now we can see the R side chains, the variable groups are down, up, down, like that. So we have this side chain very nicely straight line, but it's not really going to be the final product. So the final product is actually a helix here. So why is that? Because we had a very nice peptide bond, the covalent connection, and then it gave us a very nice linear structure. But what happened is here, N to the H, this is a hydrogen bond donor, and C double bond O, the carbonyl oxygen, is a hydrogen bonding acceptor. So donating a hydrogen bond, accepting a hydrogen bond. Now they come closer, it makes a very nice alpha helix. They could be left-handed or it could be right-handed. If you remember the DNA slide, remember you can take your arms up and the thumb goes up and the fingers circle around, so you can figure out, is it left turn or is it a right turn, using your two hands, right? So it's a good way to look at it. So now you see the hydrogen bonding makes the secondary structure. Now this has a two-dimensional shape, right, from just the 1D line. And then that's not the only one. We can also get something called the beta-plated sheet. And the difference between these two, you see this one is a single strand of single polypeptide chain, and this one has two polypeptide chains. And this is the hydrogen bonding in between the polypeptide chains here within the same one. So those details are important, and we move on. Now here, so again, going back to comparison, that's why these slides are here. I'm talking about, 2-3 lectures later we're going to talk about, but I just want to talk about that Friday because of this reason. See the similarity in the DNA. We saw the 5' 3', 3' 5', so anti-parallel very nicely docking into each other because of the geometric arrangements and the hydrogen bonding, the donor acceptor, right? Now here the same story actually. See, normally we read NASCAR, N to the C, and then C to the N, the anti-parallel. They can actually nicely interact together with the Van der Waals forces, hydrogen bonding. So not only that, in proteins there's parallel, anti-parallel. They also can have mixed ones, right? Now little bit more complex thing here. So look at the alpha carbon to the carbonyl carbon. You have a specific angle. It can go plus 180 and the minus 180, total of 360 possibility theoretically. And chi and psi here, right? And then now here the N to the alpha carbon, this one also freely rotatable. Like I said, the 6 atoms are sticking into their own planar, right? And then they are freely rotatable, but this may be governed by this side chain, right? And there are other factors too. Let's look at what it is. Now this is a Ramachandran plot. So what Ramachandran did is he studied theoretically what's the possibility if you have those psi and chi angles. So 0 to 180, 0 to minus 180. So it's rotating forward and backward, full circle in a way, right? And then you can just plot them. So when he did that, so we didn't find enormous amount of possibilities. He did find that left-handed alpha helix only restricted to this area, and right-handed alpha helix only restricted to this area. Then you have parallel, anti-parallel, and so on. So the lighter area, actually lighter green area, is more simpler side chains like alanine and glycine, and then the more restricted ones are more inner circle. So now you see the nature, the hydrogen bonding interactions and the other interactions actually governed that all mathematical possibilities are not allowed in biological systems, right? Now, if you remember the first day I was talking about, let's neglect about the Van der Waals circles, Van der Waals radius, right? Here you have 2 Van der Waals radius. Now I want to really, not the size again, not exact how many angstroms there. I just want to look at the general picture. You see the hydrogen here, the Van der Waals radius is shown here. The carbonyl oxygen Van der Waals radius is shown here. We know they cannot violate each other's surface, the area, right? Instead, this one actually does that because it's from newer research that you can see who are the people behind that, and so they have explained that this actually even cross cross the line and go to the other side, because there are other factors affecting here, right? Like the ones we just saw a moment ago. So that will restrict the possibility of existence of secondary structures, right? The alpha helixes, the left-handed, right-handed helixes, the beta sheets, and the angles, those type of information. So now from that little details, the single, the peptide bond and linear structure, and from there we goes into the alpha helixes, secondary structure because of the hydrogen bonding. We are not going to stop there, so that's secondary. Now we jump to the tertiary structure. These ones, if you remember that we talked about the sulfur bridging and ionic interactions, hydrophobic interactions, those things can bring some of the two ends come closer, and then prosthetic groups like iron atoms, Fe, and then this is actually globin protein, right? So the globin protein with the heme together, it makes the beta chain hemoglobin. It's one functional unit. Aha, now it's functional. So far we were talking about the structures. Now we get the functional protein. That's what we want, right? We need the enzymes, we need oxygen carriers, and so they are functional. They can do work. Now, the only difference I kind of see this way now, the quaternary is actually, you see this is one subunit, so you have a bunch of subunits. In this case, 4, it could be hexa, and so on, right? So that's what I want to finish today. Thanks.